Hey everyone, today I'm just talking about membrane dynamics. Right here I have this simple chart just listing the different fluid compartments of the body and then what fluid can permeate into what. So we know DTO permeates into the entire body. We know in our ECF, which is 1/3 of our body, is permeated by inulin, and then our plasma is permeated into Evans blue, which plasma makes up 25% of our ECF. And then these equations, DTO minus inulin and inulin minus Evans blue, can give us our ICF and our interstitial fluid, respectively. Osmolarity and tonicity. Whereas a solution's osmolarity is based solely on its total solute concentration, its tonicity is determined by how it affects the cell volume. So osmolarity is pretty relative. So if we look at this cell compared to this fluid, it would be hyperosmotic, whereas this fluid would be hypoosmotic to this cell. Our tonicity, or sorry, just to mention our isoosmotic, it would be the same concentration in both. For our tonicity, it always refers to the fluid in regards to the cell. So our fluid would be isotonic to the cell. Our fluid is hypotonic here because we see this smaller concentration. That means water is going to want to follow this salt gradient and go into the cell, causing cell swelling or cell bursting. And then our hypertonic occurs because this cell concentration, this fluid concentration is so much higher than the cell concentration, causing the cell to shrink. Our penetrating solutes do not play a role in tonicity. They just freely diffuse. I drew just as a simple example that in the nephron, urea just follows its concentration gradients out and in to the tubules because it just freely diffuses. In tonicity, we really only care about these non-penetrating solutes. And so I have a practice problem here to help illustrate what we've talked about. What are the final volumes if 1 liter of water is evaporated? We know it's this simple equation of solute over volume, and then I just make 2 charts of before and after the evaporation to find our answers. So we know our volumes, and we know our osmolarity because normal bodies have a molarity of 300 or a milliosmolarity of 300. Our solutes can be calculated. And then once we filled out this chart, we know our solutes will remain the same because we're only evaporating water. We know our volume is going to decrease. Our total volume is going to decrease by 1 liter, and our osmolarity actually is going to change. So that's what we need to calculate here. And once we have it here, then it will be the same for the fluid, the ECF and the ICF. And now since we have our milliosmoles and our milliosmolars, then we can determine our volumes. Thanks, guys.